Dear learners, welcome back to NPTEL online certification course on novel drug delivery systems. I am Dr. Satish Devanapelli, Assistant Professor, Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. Today, we are going to see a new topic, that is nanoparticles as a drug delivery systems. So far, we have discussed on the what are the novel drug delivery systems and their applications and other important points in novel drug delivery systems. So, today we are going to discuss on the another topic, that is nanoparticles as a drug delivery systems. So, in this, here we are going to see these are the topics of today's presentation. So, in this contents, so first I am going to discuss on the introduction on the nanoparticles as a drug delivery systems. Then further I will discuss the history behind the development of nanoparticles as a drug delivery systems. Then we will discuss on the what are the nanomedicines, and further we are going to discuss on the various terminology used in the nanotechnology. After that, we are going to discuss on the various types of nanoparticles, and at the end we are going to conclude with the various properties of the nanoparticles. So, as we know, in the recent years, nanotechnology has a tremendous impacted the field of medicine through advances in drug delivery. So, we know that if you see, there is a translation of the medicines into the nanotechnology. So, scientists are discovering or translating their products into various nano systems. For example, we can consider there are the anti-cancer drugs which people are using different nanotechnology approaches to convert into the readily available nanomedicines for the treatment of the various anti-cancers, and not only that, we can also use it for the various infectious diseases also. We can also target with this system for the treatment of various diseases. And this nanotechnology, which is not limited to the medicines, not limited to the in the drug delivery segment. So, people are using these approaches, these strategies in various sectors. In that, healthcare sector is the one segment. So, there we can use for the cosmetics and disinfections and other nutraceutical supplements. And these strategies can also used in the biological models for the development of various cell cultures, animal and tissue cultures. And this technology can also used in the treatment of various studies in the analysis, that is nothing but reporter cells, organ on chip and omics platforms. Yes, in with this technology we can also use in the environment for the wastewater treatment to purify the contaminated water. This already we have discussed in the nanomedicine. You can also use it in the drug diagnostics as well as sensors, implants and the drug delivery applications. And we can also convert the conventional pesticides into the based on the nanotechnology, we can convert and we can improve the efficacy or efficiency of the pesticides. This technology can be used in the packaging and other food processing materials. So, with this we will understand that nanotechnology is not limited to one field. So, here we can see the history. So, the Richard Feynman is introduced the concept of nanotechnology, and he is an American physicist and he received a Nobel Prize in 1959. So, he introduced one statement, that is, there is a plenty of room at the bottom. So, that means in the nanoscale there is a huge scope for the development of the medicines and other material applications. Now, we can see here the size comparison of the biological molecules with the nanosystems. So, here you can see the size range of the atoms, proteins, antibodies and the virus, the bacteria and the eukaryotic cells. Now, you can see the size of the nanosystems are equivalent to the biological macromolecules. So, the relative size comparison of nanomaterials, microbiological and other biological entity. Now, we can see the size range is almost from 1 to 100 nanometer. Now, we can see these are the various systems, carbon nanotubes and the quantum dots and the liposomes. So, this can be measured by using the scanning and transmission electron microscopy. This cannot be measured by the conventional light microscopy. Yes, you can see the bacteria and the eukaryotic cells. So, to see the nanosystems, you must use scanning electron microscopy and transmission electron microscopy. So, you can see here in this picture, the particle size of each nanosystem is almost near to the 10 nanometer. So, with this to analyze the nanoparticles, we must use the scanning electron microscopy or transmission electron microscopy. So, with that we can characterize the various nanosystems. So, this indicates the importance of nanosystems. Now, here in slide we will see the another interesting aspect of the nanomedicine. So, we can read here, nanomedicine can be broadly defined as the branch of medicine that makes use of nanotechnology for disease prevention and monitoring, intervention through new modalities for imaging, diagnosis, treatment, repair, regeneration of biological systems. So, we can, with this picture you will understand here, with these conventional formulations like capsules, tablets and the syrups and the pellets, which can converted into the nanoscale. We know that conventional formulation have limited with their solubility and they have limited in efficacy because of their permeability. So, these problems can be overcome by using the nanotechnology. This leads to, this will improve the bioavailability of the drugs. So, we know that solubility and the permeability both are the rate limiting steps for the oral drugs. So, when you use nanotechnology, this system will help to convert from the micron to nano size. So, that is the beauty of the nanotechnology. With this, we can enhance the solubility and the permeability. So, now here we are going to discuss on the various terminologies which are used in the nanotechnology. So, with this we will understand, so we can avoid the confusions between the various terminology. So, here first terminology comes the nanoparticles. The name itself defines that, which are small particles that ranges between 1 to 100 nanometer in size, that have specific physicochemical properties different to bulk materials of the same composition. So, when you have a material, right, we need to process through various nanotechnology approaches, so that that micron size of your bulk material will convert into the nano range, and definitely their physicochemical properties are different from the bulk material in terms of size, shape and surface area will vary compared with the bulk material, because we are reducing the particle size from micron to nano range, and yes, typically the size is 1 to 100 nanometer in the size. And the second terminology is nano based drug delivery. So, the name itself speaks that drug delivery, that means we are taking nano formulations and we are delivering into the target site. So, nano based drug delivery aims to target the drug payload, that is nothing but carrier system payload to the right place at the right time and at the right optimal dose. So, after administration, so by using nano based drug delivery system, your formulation or your carrier will target at the right place and at right time and at the right dose, the drug will come out at that specific site. So, that is the beauty of the nano based drug delivery systems compared to conventional nano systems. Now, the another terminology used, that is nano systems. Again, it is a type of formulation, right. So, which are also known as submicron sized particles. So, that size should be below 1 micrometer with one or more therapeutic agents that are dispersed, adsorbed on the surface or covalently bound in encapsulated vesicles or capsules or matrices. So, these are the terminology people use in the nanotechnology and in the nanomedicine platform. Now, these are some other terminologies used, that is nanocarriers. So, again we can see that which are the carriers which will carry the molecules or and deliver the substance like drugs, biologicals, diagnostics to specific sites in the body. So, the main role of the nanocarriers to carry and deliver the drugs and other biological materials to the specific site in the body. For example, you can consider taking a polymerosomes. You can encapsulate your anti-cancer drugs and you can deliver it. So, it will take your anti-cancer drug to the into the cancer cells rather than normal cells, and then there it will deliver the drug substances at right time. So, these are other terms terminology we can see here, that is nanomaterials. So, when you use different materials like lipids, we have inorganic materials, we also have polymers, right, we have a surfactants. By using of all this, we will produce the nanomaterials. So, these are the terminology, and if you would like to know more, here is the reference, you can go through and you will get all these definitions in the link. So, now these are the various categories. The liposomes, the first one, it is a lipid based formulation, and then polymer based system, that is a polymeric micelles, and the polymerosomes, and then micelles and the dendrimers. So, all these systems after converting into the nanosystems, you will get in the particle size in the range of 10 to 100 nanometer. So, now here we are going to see the types of nanocarrier systems. Yes, so based on the material, we can classify the nanomaterials into two categories. One is organic nanomaterials, second is the inorganic nanomaterials. Now, the name itself indicates that inorganic nanomaterials, nothing but which do not contain any carbon and hydrogen atoms in their structure. So, now the which comprise metallic, non-metallic elemental compounds that have weak intermolecular interactions which forms nanostructures with a high dimensionality. So, these are the few examples like nanoshells, and also we can see the quantum dots, nanoceria, iron oxide nanoparticles, gold nanoparticles, silica nanoparticles. We can also include silver nanoparticles also. So, these all are made up of the metallic and metal oxide and other type of the materials. So, which they do not contain the carbon and hydrogen atoms. Now, in another side we can see the organic nanoparticles. Now, these all materials will contain the carbon and hydrogen atoms, and that forms the hydrocarbon based molecules. So, here you can see the liposomes which contain the carbon, and polymeric nanoparticles which contain the carbon, micelles, dendrimers and the lipid nanoparticles. All lipids, polymeric based systems which definitely contain the carbon atoms. And now, the great property is that these may form by the self assembling systems. So, it forms the nanostructure by self assembling at different conditions, by applying some pressure and by applying some of the interactions with the opponent charge. So, that leads to the formation of the self assembled nanostructures. Yes, they will also utilize weak intermolecular interactions to form the cell assembles. To know more, here is the open access link. You can go to that link and you can read more information. So, yes, based on that we have classified organic and inorganic. These are continuous classification. These two comes in the organic nanocarriers and these are the inorganic. Now, these already we have discussed. It is a polymer based systems and the lipid based. Monoclonal antibody nanoparticles, dendrimers, nanospheres, then liposomes, SLN, solid lipid nanoparticles, extracellular vesicles. In the inorganic, silica, silver and the gold nanoparticles. From this you will understand the various structures or pictorial representation of the various nanocarrier systems. So, now here we will see the properties of nanoparticles. So, yes, when you compare with the conventional, the bulk materials, their physicochemical properties are completely different from the nanomaterials. Nanoparticles offer unique properties such as small size. We know we are discussing so far, yes, their size is below 1 micron and preferably 10 to 100 nanometers. So, with that we can say their particle size is very less, and yes, they have the high surface area. Nothing but when you break a large surface area particles by using the nanotechnology, and you will see the formation of the nanoparticles and their surface area will be very high compared to the bulk material which is in the micron. So, the surface area is very high. So, that is why so we convert water insoluble drugs into the water soluble drugs by increasing the surface area of the bulk materials. So, surface chemistry of the nanoparticle can be modified to enhance the drug binding. So, now here you can see the structure of the nanoparticles. So, here you can see the surface you can modify by using various polymers. So, you can improve the biocompatibility, that is possible, and you can also label with the some of the ligands which will specifically bind to the receptors. So, you can modify by using ligands for specific targeting for cancer. Yes, you can also use various drug molecules, you can load it, you can functionalize on the surface, and that will definitely control the release. And yes, you can also use various types of the fluorescent molecules, it can works as a imaging purpose. So, with this you can conclude that the surface chemistry of the nanoparticle can be modified to enhance the binding to the cells. And yes, easy to suspend in the liquids when compared with the bulk material, it will not redispersible, but after size reduction when you convert into nanoparticle, your particle will easily suspend into the liquid. With that we can enhance the solubility. And yes, compared to the conventional system, your particle may go into the deeper layers of the cells, or you can say that you will get a deep access to the cells and other organelles. So, we can target the cells, we can also target to the organs, we can also target to the organelles, nothing but we can target mitochondria and the other parts of the cells. So, that is the greatness because of their size, and they can easily penetrate into the deeper cells. And yes, with the inorganic materials, we can also use their optical and magnetic electrical properties for the imaging purpose. So, because of this, inorganic materials works as a contrasting agent in the MRI and CT scan. So, yes, by using the lipids and by using the polymers and by utilizing the inorganic materials, so we can make a nanosystem that can be utilized for the various drug delivery and the imaging application only because of their surface properties and their unique properties. So, with this, so we will summarize today's lecture. So far we have discussed on the introduction of the nanoparticle as a drug delivery system, and then we have seen the importance of nanoparticles. Then we have discussed the history of the nanomedicines, and also we have discussed various terminology used in the nano drug delivery systems, and then we have seen the types of various nanocarrier systems, and later we have discussed the properties of the nanoparticles as a drug delivery systems. So, with this we conclude today's lecture, and in next class we are going to see the other advantages and the various applications of the nanoparticles as a drug delivery systems. Not only that, we are going to discuss on each type of the nano systems with the case study. Thank you so much.